Hello, I'm Braun, the team leader.
I'm Hannah Maat, the team accountant.
I'm Lindy, Oujiri, the team manager.
I'm Nathan Rotter, the team secretary, and we are Senior Design Team 5 from the BME department. Our project is the development of qPCR assay for clinically relevant bacteria used in chemical disinfection confirmation testing, which was sponsored by Stryker. The main problem that our project aims to solve is that the current method of disinfection validating, agar plate counting, is incredibly inefficient, with high costs, low accuracy, and a tremendous time commitment. Agar plate counting could take up to weeks for certain strains of bacteria, but quantitative polymerase chain reaction, or qPCR, only takes a few hours. By replacing the current method, we would be able to more efficiently and accurately count bacteria remaining following disinfection.
qPCR is a technique that can be used to quantify an unknown amount of DNA in a sample. The process starts with a very small amount of DNA mixed with primers or short DNA sequences that bind to the target DNA template strand. Additionally, fluorescent probes are added to the reaction mixture. These probes emit light once they bind to the DNA sequence. Once placed in a well plate, the mixture undergoes a series of cycles in a machine called a thermocycler. These cycles involve repeated heating and cooling of the sample, which splits the DNA strand and allows it to be copied exponentially. The copying triggers the release of fluorescent molecules, which produce a signal that can be measured by the machine, allowing us to quantify how much DNA was in the original sample.
The main focus of this project was to develop a protocol to test the efficiency of replacing agar plate counting with a qPCR assay as the validation method for medical device disinfection. Here, we aim to develop unique primers and probes targeting the bacterial species used in disinfection validation, which are E. coli, P. aeruginosa, Klebsiella ammoniae, and Staph aureus. These unique primers are designed so that the sequences targeting each species will not interact with the others while testing multiple species at a time in a process called multiplexing. First, our team designed reproducible protocols for agar plate counting and qPCR standard curve generation. Then, we developed a method for creating and testing unique primer sequences for each bacterial species. Lastly, we performed exploratory testing on potential primer sequences using cyber green dye. Results revealed that we obtained preliminary success in primer sets for 3 out of the 4 tested species, with the primer successfully amplifying the target DNA.
Our recommendations moving forward include creating and testing more forward and reverse primer and probe sets that will successfully tag and amplify sequences unique to each strain of bacteria. The ultimate goal of the project would be to achieve multiplexing capabilities, allowing each strain to be tested in one trial of qPCR that would generate separate amplification curves. This method has the potential to lead to the implementation of a new industry standard for validation of disinfection methods that is more time and cost efficient. We would like to thank our advisors, Dr. Bruce Lee and Dr. Mohammed Rizwan, as well as the Stryker team, Dr. David Jang and Isaiah Davis. Lastly, we would like to thank our support staff here at Michigan Technological University, Fatima Razaviamri, David Dixon, Dr. Amy Marsau, John Romanowski, and Dr. Yang Zhang. Thank you very much.